Hello dear students, in this video we shall be studying the structure and functions of endoplasmic reticulum. Let's get started. The endoplasmic reticulum, abbreviated as ER, is a dynamic and extensive network of interconnected membranes found within the cytoplasm of eukaryotic cells. Structurally, the ER exists as a continuous membrane system comprising flattened sacs called cisternae and a network of interconnected tubules. This extensive network can constitute a significant portion of the total membrane in a cell, highlighting its importance. The lumen, or the internal space enclosed by the ER membrane, is a distinct compartment separate from the surrounding cytosol. The ER is typically divided into two functionally and structurally distinct regions: the rough endoplasmic reticulum (RER) and the smooth endoplasmic reticulum (SER). The RER is characterized by the presence of ribosomes attached to its cytoplasmic surface, giving it a rough appearance. The SER lacks attached ribosomes, giving it a smooth appearance. It is typically more tubular in structure compared to the flattened sacs of the RER and is often found more towards the periphery of the cell. Structure of RER, primarily composed of flattened cisternae, which are often stacked upon one another. The membrane of the RER is continuous with the outer nuclear envelope. Functions of RER. Protein synthesis. Ribosomes on the RER synthesize proteins that are translocated into the ER lumen or embedded within the ER membrane. Protein folding and modification. Within the RER lumen, proteins undergo folding with the help of chaperone proteins. Various post-translational modifications, such as glycosylation, addition of carbohydrate chains, also occur here. Quality control. The RER has a quality control system to ensure that only properly folded proteins proceed to their destinations. Misfolded proteins are retained and targeted for degradation. Transport. Proteins and lipids synthesized and modified in the RER are packaged into transport vesicles that bud off from the RER and move to the Golgi apparatus. Membrane synthesis. The RER also synthesizes phospholipids, which are essential components of cellular membranes, including its own. Functions of SER. Lipid synthesis. The SER is involved in the synthesis of various lipids, including phospholipids, steroids such as cholesterol and steroid hormones, and oils. Detoxification. In liver cells, the SER contains enzymes that detoxify lipid-soluble drugs, metabolic waste products, and harmful chemicals by converting them into more water-soluble compounds that can be easily excreted from the body. Calcium storage. The SER, particularly specialized forms like the sarcoplasmic reticulum in muscle cells, is a major storage site for calcium ions. The release and uptake of calcium from the SER play critical roles in various cellular processes, including muscle contraction and cell signaling. Carbohydrate metabolism. In some cells, the SER is involved in carbohydrate metabolism, such as the breakdown of glycogen into glucose in liver cells. These were various functions of RER and SER we have explained with their 3D structures. Don't forget to like and subscribe. See you in the next lecture. Goodbye.